1. Genetic inheritance and genetic control. Subtopic 1.1 Mendelian genetics. In this subtopic, we are going to look at monohybrid inheritance, dihybrid inheritance, and extension of Mendelian genetics. Mendelian genetics. There was once an Austrian scientist named as John Gregor Mendel that had discovered basic principles of heredity through experiments using garden pea plants. He is known as the father of genetics. So before going further into this subtopic, we need to understand a little bit on the pea plant reproductive organs. In this diagram, it shows a pea plant reproductive organs. Each pea flower has both sex organ. The male sex organ is the stamen. The stamen consists of anther and filaments. For the female sex organ, it is the carpel. The carpel consists of stigma, style, and ovary. In nature, pea plants usually self-fertilize. Mendel carried out two types of pollination amongst the pea plants. The first type of pollination is self-pollination, and the second one is cross-pollination or hybridization. Self-pollination occurs when pollen released from anther lands on the stigma of the same flower or another flower of the same plant. Sperm from the stamen fertilize ova in the carpel. For cross-pollination or hybridization, it involves fertilization that occurs between different plants. The transfer of pollen from flower of one plant to stigma of flowers of another plant. So it means it involves two individual plants of the same species. A true breeding plant is a plant variety that when the plant self-pollinates over many generations, all of their offspring have the same trait. For example, a plant with purple flower with the genotype homozygous dominant is true breeding if seed produced by self-pollination all give rise to offspring with the same phenotype which produces also purple flower that is also homozygous dominant. Mendel discovered the basic principles of heredity by breeding garden peas in carefully planned experiments. The advantages of using pea plants for the genetic study are: first, there are many varieties with distinct heritable characters such as, for example, flower color and seed color. Second, each character have variations called traits. For example, for the character flower color, it has two different traits, which is purple or white flowers. For the character seed color, it has two different traits also, which is yellow and green seed color. Third, pea plant has short generation time. Next, pea plant can produce large number of offspring from each generation. Next, mating of plants can be controlled. Next, pea plants have both pollen-producing organ, which is the stamen, and egg-bearing organ, which is the carpel. In Mendel's experiment, he prepared two true breeding parent plant, which is the pea generation through self-pollination. The pea generation has contrasting characteristics. For example, Mendel prepared true breeding parent plant that has purple flower and true breeding parent plant that has white flower for the monohybrid cross. The genotype for the purple flower plant will be capital P, and the genotype for the white flower plant will be small p, small p. He then cross-pollinated between two parent plants to produce F1 generation. Cross-pollination is also known as hybridization. Basically, hybridization involves mating or crossing of two true breeding varieties. From this hybridization, it produces F1 generation. F1 generation are the hybrid offspring of the P generation or the parental generation. F1 hybrids will then self-pollinated and produces F2 generation. This diagram, it illustrates a genetic cross between a true breed purple flower plant and a true breed white flower plant. The methods are: number 1, Remove stamen from purple flower plant. When the stamen are removed from this flower, this plant will become the female parent with the genotype homozygous dominant. Capital P, capital P. So this will become the female parent. Second, transfer pollen from stamen of white flower to carpel of purple flower. So this means the white flower plant will become the male parent with the genotype homozygous recessive. Small p, small p. So this is the male parent. Next, pollinated carpel then maturate into pod. Next, seeds from the pod are planted. The seeds will then grow into F1 generation, and all of the offspring of F1 generation will have purple flower. The result is the same for the reciprocal cross, which involves the transfer of pollen from purple flower to white flower plants. So it means that for this time, the male parent is the purple flower plant and the female parent is the white flower plant. In the parental generation, the cross between two breeding purple flower parents with the genotype homozygous dominant to true breeding white flower parents homozygous recessive produces F1 generations or F1 hybrids that has all purple flower. So this is 100% heterozygous with all purple flower. For the F1 generation, they will then undergo self-fertilization or cross-pollination and produces F2 generation. In the F2 generation, it shows in the diagram here, it produces both phenotypes. Okay, the offspring produces both phenotype, purple and white flower in the ratio of 3:1. Mendel observed the same patterns of inheritance, which is 3:1 phenotypic ratio in the F2 generation in the 7p plant characters. Each character is represented by two traits. For example, for the character seed color, the crossing between parent that has yellow seed to a parent which has green seed produces later in the F2 generation, the offspring will have phenotypic ratio 3 yellow and 1 green seeds. Based on Mendel's quantitative analysis of F2 plants, he proposed two fundamental principles of heredity. The first one is the law of segregation, and the second is the law of independent assortment. Monohybrid inheritance and the law of segregation. Mendel performs breeding experiment which involves only a single or mono character, for example, flower color, to derive the law of segregation. The hybrid produced are monohybrids. These monohybrids are basically heterozygous for one character. Monohybrid. Monohybrid is an organism that is heterozygous with respect to a single gene of interest. This gene will express a certain character. Monohybrid results from a cross between parents that are true breed for different alleles. For example, parents of genotype homozygous dominant P crosses to a homozygous recessive parent with the genotype small p produces a hybrid which is a monohybrid with the genotype heterozygous P. Mendel's first law, which is the law of segregation. This law states that the two alleles in a pair of homologous chromosomes segregate or separate from each other into different gametes during gamete formation. This segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis. Thus, an egg or a sperm gets only one of the two alleles that are present in the somatic cells of an organism. Basically, this law explains how two alleles of a gene end up in different gametes during meiosis. In this diagram, a pair of homologous chromosomes that carries two different alleles for the gene that controls plant height. This one. Okay, so it has the dominant allele capital T and the recessive allele small t. During metaphase 1 of meiosis 1, the homologous chromosome align at the metaphase plate. Then during anaphase 1, the homologous chromosome segregate or separate into two different daughter cells. This separate the two alleles into different cells that later at the end of meiosis 2 produces gametes that carries different alleles. Punnett square. Punnett square shows the possible combinations of sperm and eggs during fertilization. Punnett square also predicts the results of a genetic cross between individuals of known genotype. From this, you can predict the genotype of the offspring. Next, Punnett square can also predict the genotypic ratios and also the phenotypic ratios of the various offspring from a cross of two individuals. Let's take for example the self-pollination or cross-pollination that occurs in the F1 generation. So you have to write the genotype for the male parents. The male parent would have the genotype heterozygous. Same goes to the female parents, would also have the genotype of heterozygous. So both of these individuals would have purple flower. And then for the next step is that you have to determine the genotype for both of the parents. For the gamete for these two individuals, 50% of the gamete would have the dominant P allele, and another 50% of the gamete would have the recessive small p allele. So in order for you to write the gametes, you have to write it in a circle to indicate that these are gametes. Same goes to the female individual. Half of the gametes would have the dominant P allele, and the other half of the gametes would have the recessive P allele. So you write here gametes. Next, you need to draw a Punnett square. So you just draw a table like this. If this involve two different gametes for both individuals, so you have to draw basically three rows. Okay, so the first row is for you to write the gametes for the male and the female parents. Okay, so for the male parents, half of the gamete would have the dominant P allele, and half of the gametes will have the recessive small p allele. Same goes to the female parents. So half of the gametes would have the dominant p allele, and another half of the gamete would have the small p allele. Okay, so you have to combine these the gametes together during fertilization. So the second row, the second column here, you have the combination between the alleles in the sperm and also alleles in the eggs. Okay, so this produces homozygous dominant individual. So the phenotype for this individual would have purple flower. Okay, so the next individual here, you have to combine the dominant P allele with the recessive small p allele, and this also produces purple flower. And then next, you do the same, the combination between the sperm that has dominant P allele with the eggs that has the recessive small p allele. This also produces purple flower. Next, what you do, you combine the sperm that has the recessive small allele with the eggs that has the recessive P allele, and this also produces white flower. Okay, so next you have to determine the genotype for the offspring produced. So in the box here, okay, these are the F2 generation. Okay, so the genotypic ratio for the F2 generation would be 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. And the phenotypic ratio for the F2 generation would produce, these two genotype produces 3 individuals that has purple flower and 1 individual with white flower. Okay, so that's how you construct a Punnett square. So this is basically a Punnett square. An individual with a dominant phenotype could be either homozygous dominant or heterozygous. For example, you see a plant with a purple flower. You know that the purple flower is a dominant trait, but you don't know the genotype of this plant. The plant can either be a homozygous dominant plant or a heterozygous individual. Okay, so the genotype is unknown for that individual. So what you do is that you do a test cross. What is a test cross? Test cross is used to identify the genotype of the individual with the dominant phenotype. So the method of test cross is breed the mystery individuals with a homozygous recessive individual. So the genotype for this homozygous recessive individual, for example, would have the small p recessive allele. If any offspring display the recessive phenotype, so therefore it means that the mystery individual must be heterozygous. A plant with a dominant phenotype, as you can see, for example, in this diagram, a plant with purple flower, you don't know whether the plant is a homozygous dominant or heterozygous. So for this purpose, you need to cross the plant with a homozygous recessive individual that has white flower. So there are two possible outcomes. If the unknown individual is a homozygous dominant individual, all of the offspring produced is a heterozygous and would produce all purple flower. But if the individual is heterozygous, 50% of the offspring would produce purple flower and another 50% of the offspring would produce white flower. Dihybrid inheritance and the law of independent assortment. So what is a dihybrid? Dihybrid is an organism that is heterozygous with respect to two genes of interest. So it means that this dihybrid is a heterozygous individual that we studied for two different characters. Dihybrid results from a cross between two true breeding parents that differs in two characters. These true breeding parents are homozygous for two different alleles. The crossing involves two characters that is controlled by two genes. For example, crossing two true breeding pea varieties which differ in two characters. For example, a parental cross between an individual with yellow round seeds. This individual is basically homozygous dominant for both alleles. And another individual with green wrinkle seed which is homozygous recessive for both alleles. As you can see from the genotype here, there are two genes that govern the expression of two different characters. The first gene here is controlled by the Y allele. So the Y allele will determine the seed color. And as for the second gene, which is governed by the R allele, it expresses the seed shape. Basically, for dihybrid cross, it involves two different characters that is controlled by two different genes. Example here, you have the gene for seed color, which is composed of or determined by the dominant Y allele and the recessive Y allele. The dominant Y allele would produce yellow colored seed, and the recessive small Y allele would produce green seeds. For the second gene, the gene for the seed shape controls by two different allele. The dominant R allele, which expresses round seed shape, and for the recessive allele produces wrinkle Seed shape. Parental generation. The first parent that is homozygous dominant for both genes with the phenotype yellow round seeds crosses with another parent that is homozygous recessive for both genes with the phenotype green wrinkle seeds. 100% of the gamete for the first parent would have one dominant allele for both genes. Each gamete will have one dominant Y allele and one dominant R allele. And 100% of the gamete for the second parent would have one recessive allele for both genes, which is every allele will have one recessive Y allele and one recessive R allele. Fertilization between gametes from both parents produces F1 generation that is heterozygous for both genes. Therefore, 100% of the F1 generation would have the dominant phenotype, which is yellow round seeds. The F1 dihybrids were allowed to self-pollinate to produce F2 offspring. In the production of gametes for F1 dihybrids, the Y alleles for seed color segregate independently of the R alleles for seed shape. For this heterozygous genotype with 4 different alleles, 4 different gametes will be produced. The 4 gametes produced are capital Y, capital R, capital Y, small r, small y, capital R, and small y, small r. Each of these type of gametes will be produced in the amount of 25% each or 1/4 each. Because these are gametes, you have to draw a circle around each of the gametes. From the Punnett square, 16 probable ways of the alleles can combine to produce the F2 generation. From the Punnett square, the phenotypic ratio of F2 generation would be 9 yellow seeds, round seeds, to 3 green round seeds, to 3 yellow wrinkle seeds, to 1 green wrinkle seeds. Using a dihybrid cross, Mendel developed the law of independent assortment. The law basically states each pair of alleles from one gene segregates and assort independently with other pair of alleles from another gene during gamete formation. This law applies only to genes on different non-homologous chromosomes or those far apart on the same chromosome. Basically, both of the Mendel's law of heredity can be observed during gamete formations that occur during meiosis, which is the law of independent assortment and also the law of segregation. For the law of independent assortment, the non-homologous chromosomes that carries alleles of different genes will assort independently of each other at the metaphase plate during metaphase 1. There are many possible combinations of alignment of these non-homologous chromosomes along the metaphase plate, which is depending on the number of chromosomes an organism have. For this example, you will see that there are 2 possible combinations of the assortment for the non-homologous chromosomes. After all the homologous chromosome has aligned at the metaphase plate, we can apply the law of segregation. During anaphase 1 of meiosis 1, the 2 alleles of each gene on the separate chromosome will separate or segregate into 2 different daughter cells. At the end of meiosis 2, each alleles of each genes will end up in different gametes. Each of the gamete produced therefore will have different combinations of allele from each genes. These are the gametes of the F1 generations. The fertilization of F1 gametes produces F2 generations with a phenotypic ratio of 9 to 3 to 3 to 1. Basically, fertilization recombines allele from each gene at randoms. Each individual gets random recombination of alleles for each genes. So that's why for the F2 generation, you will see that there are many new phenotype produced that is different from the F1 generation and also the original parental generation.